A new crime scene has been unveiled down in Soho, New York. We have arrived to the scene to investigate. After intense investigation, we have come to the conclusion that our victim has been brutally stabbed to death. It is important that we collect a sample of DNA from the scene to find out who our suspect is. Our best source for DNA will come from the victim's blood. To do this, we collected a sample by using a pipette to carefully collect a selection of DNA, and we placed it into an airtight sealed container to prevent unwanted environmental influences. We are now traveling straight into the lab to dive into this crime scene. Buffer concentrate, distilled water, and agarose powder are poured into the beaker. It is all then mixed and stirred until the agarose is dissolved. Now the agarose is heated on a hot plate. Once heated, the agarose is poured and solidifies into agarose gel. A comb runs through the gel to even out the surface. The solution containing the DNA fragments are loaded into the wells of the gel electrophoresis apparatus. Now, the DNA strands are then cut by restriction enzymes. It is important to know that DNA, also known as deoxyribonucleic acid, is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The 4 types of nitrogen bases are adenine, thymine, guanine, and cytosine. The order of these bases is what determines the DNA instructions or genetic code. Now, the electric current is run through the gel and is used to establish an electrical field around the fragments. Over a period of time, the fragments of DNA will migrate through the gel towards the electrode of opposite charge. We can now compare the different strands of DNA in the agarose to see which two are identical. The strand that matches the marker DNA reveals who is guilty.